I think, one big departure from the typical graph that we see with energy systems, right? We see phosphocreatine is used in the first 10, 20 seconds, and then it goes to zero, and then it's not used anymore. That's not true. It keeps being used all the time.
I thought a good place to start would be the evolution of these traditional views. So, aerobic, anaerobic, creatine phosphate. Could you tell us why that model has sort of fallen away or lost relevancy in the new world?
Yeah, I think it's a good starting point. So what I learned when I started reading about training and about bioenergetics was those three energy systems, right? And obviously the main goal of those energy systems is to put ATP back together, adenosine triphosphate, which is the energy currency of the body, one of them. And those energy systems are there to just put it back together, because we essentially break it down when we contract muscle, or essentially any energy you require in the body is going to go through those pathways, whether it's maintaining cellular structures, creating new proteins, or again powering mechanical work, so muscle contractions, for example. And so the traditional model that I was introduced to, and a great book that I read early on, was Ultimate MMA Conditioning from Joel Jamieson. And I think you could take the MMA out and call it Ultimate Sports Conditioning. I think it was a good overview. And the beginning about energy systems, when he describes the three systems, is something that was always fascinated by, and trying to understand the mechanistic aspect of how the body works. I definitely think that you don't need to understand all those things in detail to be a good coach, right? But if you're curious, then trying to tie together what's happening in the field and then what's happening inside the body is super interesting. And so the let's call it the old model of bioenergetics is that you had that traditional graph with those three energy systems that sequence each other in time, where you typically have the creatine phosphate that seems like it's full at the beginning, and then it goes down over 10, 30 seconds, and then it flatlines, and then it stops being used. And then you have the glycolytic pathway, which essentially the breakdown of sugar, and that creates ATP as well, or recycles ATP as well. And then you have the aerobic component that would typically take a longer time to start rising, and then it would be the main system after a period of time. And so that's kind of the traditional, or the old model of bioenergetics. And what I started understanding when I started following Evan Pycon many years back now is that those energy systems aren't separated in time, or nowhere as much as they could be understood to be in that graph format. And one of the things that Evan put forward, and everything well documented in the literature, and papers that have been out for 20, 30 years. It's not like it came out last year and it's a brand new thing. We've known that for a while. And the main thing is that all energy systems are active within 150 milliseconds, I think, of a muscular contraction. So it all turns on right away. That's the first thing to understand. So that would be one major departure from the old model, where we sequence them in time. And there was also this view of training, and that's something that was put forward in Joel's book, is that essentially, depending on the time domain of your training, you're going to target different systems, right? So if you do really short intervals, you're going to target the phosphocreatine system. If you do 30 seconds to 2-3 minutes, you're more in the glycolytic pathway. And then if you do the longer stuff, you're more in the aerobic pathway. And so we have those three systems. The first two that are coined anaerobic, because there's technically no oxygen in the chemical process of those pathways. And then there's the aerobic system on the other side, which obviously has oxygen as its main, or as an important contributor to the process. But again, all those systems work together. They all turn on pretty much instantaneously as soon as you start any kind of muscular contraction or mechanical work. And what I found after this as well is that they look separate on paper. So if you write down the equations, obviously they're different equations, they're different systems, quote unquote. But once you start paying attention to where they exist, those energy systems, and how they work, they actually all work together in one way or the other. And so I think it's interesting to think about where those those energy systems exist, which is inside the muscle fiber, right? Inside the muscle cell itself. And then inside the cell, you have things called myofibrils, which are the contractile elements of the fiber, and then you have the mitochondria, which is often coined the powerhouse of the cell, right? And from more recent research, we know that it's not only the factory for energy inside the cell, it seems to also be the power grid. So there's a lot of energy transfer that goes through the mitochondrial system, and we see mitochondria as like a little floating object inside the cell, but it's actually more like a network. And there's imaging that we have now that really shows how it's all part of the architecture and the structure of the cell itself. So it's literally like a power grid holding everything together, moving energy from one side to the other. And obviously, if we take those contractile elements in the middle of the cell, they're going to need a lot of ATP in order to contract, relax, right? And especially, typically, it's actually the relaxation phase that requires a lot of ATP, because the contraction happens through the calcium, the interaction of calcium with is it the troponin, I think, that kind of moves aside, and the myosin head binds and then pulls. But without going too far into this, coming back to the energy side of things, and I'll start with the creatine phosphate pathway, or the phosphocreatine energy system. So we call this one alactic anaerobic. Alactic because there's no lactate produced or involved, and anaerobic because there's no oxygen involved. And the way I like to frame the phosphocreatine system is like a bathtub. So when you're resting, the bathtub is full, and obviously the plug is in, and the tap is running just a little bit. It's just kind of dripping, right? And this is kind of your basal metabolic rate. So it's just the energy that you need to keep things going, but there's no major work being produced because you're at rest. And essentially, it's a buffer. It's an energy reserve that you have inside your cells. And as soon as you start an effort, the first thing that it's like pulling the plug out of the bathtub, and then that bathtub starts draining. And the more intense the effort, the bigger the drain hole, right? And so you have that reserve, you have that bathtub that starts emptying itself as soon as you start exercise. But then what happens? We need to fill up that bathtub, because we know that phosphocreatine is the fastest means of putting ATP back together. And we have to go through that pathway most of the time in order for it to work. Otherwise, other pathways are a little bit slower, quote unquote, especially the aerobic pathway. And there's also, if we go back to this idea of where things happen, there's also the fact that ATP that's produced inside the mitochondria through the oxidative pathway cannot diffuse from the mitochondria all the way to the contractile elements in the cell. The distance is too far. It's just not efficient. So the body developed something called or evolution, I guess, made happen something called the phosphocreatine shuttle. And actually, if you and this is nothing new. You can dig onto PubMed and you'll find all that information. What happens is that ATP is produced inside the mitochondria through the oxidative process. That ATP is then used to put phosphocreatine back together, because we had phosphocreatine, it broke, right? Because we put the phosphate back onto the ADP to make ATP again. But then we need to put phosphocreatine back together at some point, right? That actually happens inside the mitochondria with the ATP that is produced through the aerobic pathway. And then that phosphocreatine that's been put back together inside the mitochondria then gets diffused out towards the contractile elements. And this is called the phosphocreatine shuttle. So already here, we can see that if there's no oxygen available, there is no recomposing of the phosphocreatine system. So that bathtub essentially needs to be filled up by opening the tap. And the tap is the energy that comes from the mitochondria itself, right? So already we can take that anaerobic alactic system and realize that, sure, we have that reserve at the beginning, and then you start exercise and it starts going down. And the way you have to understand it is that Phosphocreatine is always the intermediate between the oxidative system and the muscle contractions, meaning that even though after if you go into hard effort right off the bat, for example, the bathtub, the water level is going to go down really fast, but then the drain is fully open and the tap is fully open. So even though the concentration level of phosphocreatine in the cell is low, the flux is really high because we have to go through that bathtub in order to keep using the ATP that was made in the mitochondria. And that's, I think, one big departure from the typical graph that we see with energy systems, right? We see phosphocreatine is used in the first 10, 20 seconds, and then it goes to zero, and then it's not used anymore. That's not true. It keeps being used all the time. And then obviously, if you stop exercising, it's like putting the plug back into the bathtub, and now it fills back up thanks to what? The oxygen, the mitochondria. And then when it's full, the tap closes, your bathtub is full, and you're ready for the next bout of exercise, right? So already we can see that phosphocreatine and oxidative work together, and they might as well be one and the same. Obviously, chemically, if you write down the equations, they're not together, but functionally inside the body, they work as a pair. So that was one big realization that I had learning from Evan. And then if we start to add in the glycolytic pathway, glycolysis happens inside the cytosol of the cell, so inside the kind of the body, inside the main space inside the cell. And it does produce ATP pretty quickly, but then comes with it the byproduct of glycolysis, which is typically lactate. There's a balance between lactate and pyruvate, but we know now that it's mainly lactate and then a little bit of pyruvate. And then depending on the capacity of the cell, it's either going to be recycled fully in the mitochondria, or if it can't fully be recycled by the mitochondria, it's going to be put back into the blood, into the circulation, to go and get used somewhere else. Our brain uses lactate, our heart uses lactate, every muscle can use lactate, because essentially the mitochondria takes lactate and then it's broken down through the Krebs cycle, and then that feeds the electron transport chain, right? So if we go back to the mitochondria for a second, inside the mitochondria, we have those kind of two main stages. We have the Krebs cycle that happens inside the mitochondrial matrix. So it's going to take, to simplify, it's going to take pyruvate or lactate, or whatever comes out of beta oxidation, so the breakdown of fatty acids, and it's going to feed that into the Krebs cycle. And then from this process, we're going to get a little bit of ATP. We're going to get some CO2. This is where CO2 is produced inside the metabolic process. And we're going to get the NADH and the FADH2. And those two molecules, we can think of them as electron transporters, right? And then those feed into the electron transport chain, and then that creates the proton gradient, and then that generates ATP with oxygen that comes in at complex 4 of the respiratory chain. And then, so going back to glycolysis, that happens inside the body of the cell. So now we come out of the mitochondria. We're back into the main body of the cell. Like I was saying before, and if we start working in different intensities, it might be helpful. So typically over the intensity spectrum, I would say there's 4 different intensities of exercise on the endurance side of things. There's low intensity, medium intensity, high intensity, and very high intensity. And we can come back to those later and break them apart, and introduce concepts like thresholds. But for now, let's just think that there's 4 levels, okay? That's the simplest way to think about it. So when you're at level 1, when you're at low intensity, your lactate production through glycolysis is fairly low. It's always going. Even right now, if we prick your finger, you're, I don't know, maybe at 1 or 1.5 millimoles of lactate. So glycolysis is always active. It's wrong to say that there's no sugars being used at low intensity. They are used, but obviously less than when you're at higher intensity. But anyway, glycolysis is happening at low intensity, but essentially you have more mitochondria than you need to recycle the lactate that's produced. And so what happens with the lactate in the blood, it either stays the same or goes down, right? So let's say you're at 1.5 millimoles measured in the blood right now at rest. I get you to start exercising at a low intensity. After 2 or 3 minutes, I prick your finger again, and you might actually come down to 1, maybe even 0.5, right? Because you're producing very little lactate inside the cell because you're preferentially using fats at that point. And so your cell is actually going to go and pull sugars from your blood to keep feeding that mitochondria, right? So that's why lactate actually drops typically at low intensity. compared to baseline resting levels. So that's why, depending on the intensity, different things happen with that pyruvate lactate inside the cell. And then now if you go to medium intensity, so that would be between the two thresholds, right? That would be a sustained effort somewhere between, say, 4 and 7 out of 10 in terms of RPE. So it's hard in the legs. It's starting to be sustained on the breathing side, but it's still manageable, still something you can hold for quite a while. And what happens there is that you've essentially saturated your mitochondria. It can't recycle all the stuff that you produce inside the cell. So what's going to happen is you're going to recycle everything that you can through the mitochondria, okay, because that's the most efficient pathway. And then the excess that you're producing is going to go into the blood. And that's what we measure when we do a lactate sample at medium intensity. So you were at 1.5 at rest, you went down to, say, 0.8 at low intensity. Now it's going to start rising back up. Maybe you go to 1.5 when you're at, say, 4, 5 out of 10 in terms of difficulty. Maybe you go to 3, 4 millimoles when you go to 6, 7 out of 10. And then if we go now over to the high intensity, so kind of the third level of intensity that we can use as a model. So the high intensity VO2 max, zone 5, whatever word you want to use. Now you've saturated your mitochondria locally, so we can't recycle everything that's produced, and even what you put out in the blood can't be recycled properly everywhere else because your global capacity is just not sufficient anymore. And this is when you kind of have that runaway effect where lactate starts increasing fast, heart rate increases fast, VO2 increases fast, and you trend towards task failure. Typically, you have to slow down or you have to stop. And so again, if we go back to the energy systems, glycolysis feeds directly into the mitochondria. So glycolysis feeds directly into the aerobic system. So again, nothing works in isolation. And if we want to take a 30,000 foot view now of everything that I said, and if things were a little bit technical, I encourage you to listen to it again, maybe go read some stuff on it. I didn't understand any of this when I started looking into it, and it just takes repetition like anything else to get it. But the 30,000 foot view is that nothing works in isolation. All those systems work together. And so I feel like the model of training energy systems is not as useful as thinking about those 4 levels of intensity: low, medium, high, very high intensity. And that this model, and again, all models are wrong, but some are useful. And I find the intensity spectrum model a lot more useful when it comes to talking about endurance and talking about training than just thinking about energy systems, because again, they all work together. And obviously, like you saw, at different levels depending on the intensity that you're at. But it's actually not practical, I find, to think about, okay, I'm going to train that energy system with that time frame or that energy system with that time frame. So coming out actually of the kind of mechanistic physiology and just focusing on, okay, am I at low intensity, medium intensity, high intensity, or very high intensity? That tells me what is going on, tells me what adaptations I'm going to get, tells me how to structure my training, tells me how to organize different phases of training depending on when I am in the season. And it's a lot more useful to think and to program. So understanding all the physiology is cool. It's if you're curious, it's super interesting. But you have to tie it back in with the intensity spectrum, with the thresholds, with those 4 different levels to make it really practical and useful.